Hello, can somebody hear me? Confirm that you hear me?
Yes.
Yep.
Great, thank you. Let me share my screen here. A little slow here. There we go. You should be able to see my screen now. Are there any questions before we begin? All right, I'm not hearing anything, so I'm assuming there's no questions. Today is May 26th, and we're supposed to be finishing chapter 14 and starting chapter 13. We're a little bit behind schedule. I think we'll finish chapter 14. I don't know if we'll get started with chapter 13. There is no lab today. I will be logging in for 15 minutes to answer questions, but after that, after the last questions, I'll log off at 6:45. Any questions about what we're doing? All right, last time we had talked about the severity or duration of the disease, and we discussed both latent diseases and herd immunity. Any questions about either of those? So when we're talking about disease, it's important to realize that there are patterns to disease. Normally, there's a specific sequence of events associated with an infection by a microbe, and then the subsequent disease the patient gets. For example, a disease-causing microbe has to come from a reservoir. A reservoir must exist, and this is where the microbe comes from before it infects a patient. The pathogen must be transmitted to a susceptible host. The pathogen then has to invade the host and multiply within the host. Furthermore, the pathogen has to injure the host before there can be disease. Without injuring the host, we don't have disease. That would be an asymptomatic, disease if the pathogen does not injure the host significantly. And as you know, there are many asymptomatic diseases, including a lot of young people with COVID-19. The illness ends when one of two things happens. The host immune system could destroy the pathogen, then the illness will end. The illness can also end if the pathogen kills the host. Those are the two ways an illness can end. Any question about either of those? There are a number of predisposing factors that can make the body more susceptible to, and therefore affect the occurrence of disease. For example, females have a shorter urethra than males. Therefore, they have a higher incidence of bladder infections. There are inherited traits such as the sickle cell gene, which can increase your chance that you'll come down with another disease. Another trait which isn't exactly inherited, although it's partially inherited, is diabetics are much more likely to come down with a disease than a patient who's not a diabetic. The climate and the weather can affect the occurrence of disease. As we all know, colds and flus are more likely to spread in the wintertime. And there's actually a reason for that. We won't go into it in any detail. I mean, you don't need to know the reason, but There's less UV light in the sun, and the UV in the wintertime, there's less UV light in the sun, and the UV light destroys the genome of the virus, as well as colds and flus are actually stabilized in cooler weather. They're more likely to die out in a warmer climate. And actually, this is something you might come to learn if you ever come down with a COVID-19 infection, and that is increase the temperature in your room or your house. And that is because COVID-19 also is affected by the temperature. And the warmer it is, the what do you say, shorter the lifespan of the virus will be. Any questions about any of that? Fatigue also affects the ability of someone to come down with the disease. Someone who's fatigued is much more likely to come down with the disease than someone who's rested. Age increases the incidence of disease. In most diseases, let's see if I can show this. In most diseases, it's both the young and the old that are at greatest risk for getting a disease. And so when we look at the incidence of most diseases, and so this would be the incidence of disease on the y-axis, and then the age on the, see if I can write this, not very good, age on the x-axis. Did I say incidence on the y-axis? And that is most diseases are much more likely to occur at a very young age and then decrease. And then when a person is either an older child or a teenager or a young adult, the incidence of disease will be low. And then with age, it increases again. And some diseases, actually, it increases quite a bit at old age, okay? Any question about that? So most diseases take this incidence. The very young get the disease and the very elderly. Now with COVID-19, it's actually much lesser of this degree, and then it's like this for the elderly, meaning that only very young, like newborns, are at increased risk for COVID-19, and then the elderly are at increased risk for COVID-19. Now I should state this is the original COVID-19. The later variants of COVID-19 have seen an uptick in the young and not much of a decrease in the elderly. So COVID-19 is, with each new variant, starting to become more like a normal disease. It's not there yet. It's still the young or is not as much of a risk as the elderly. Any questions about any of that? And I guess what we could say is your chance of dying of the flu, for example, is much higher at very young ages. And then it's also higher at very old ages. Let's see, I don't think I need that, so I'm going to close that. No, I don't think I need the syllabus because we're just going to be having the lab later today. And it'll just be a question and answer session. All right, lifestyle can affect your susceptibility to a disease. Smokers are much more likely to come down with a respiratory disease. And that's because smoking damages the mucosa in the respiratory system. And so they're more likely to come down with the respiratory disease. There's other lifestyles that can affect whether or not you come down with a disease. And oh, I can't think of another good one. Obviously, tennis players are much more likely to come down with tennis elbow. And that's not an infectious disease, but it's another disease caused by lifestyle. Maybe somebody can give me an example of a lifestyle disease, because I'm not thinking of one off the top of my head, but I know smokers much more likely to come down with a respiratory disease. Chemotherapy is much more likely to predispose you to an infection. Chemotherapy weakens the immune system as well as weakening your other body defenses. And that makes you more susceptible to coming down with another disease. Any questions about any of that? I should mention that students often don't get enough sleep because they're studying and working so much, and that makes you more at risk for coming down with an infection. So when you get a chance, try to sleep. The disease tends to develop in the same way. Once the host defenses are breached, disease development follows a certain sequence. And this sequence tends to be similar for both acute and chronic diseases. First, you have a patient who's infected. And from the time they're infected till they have the first signs or symptoms of disease, we call that the incubation period. So the incubation period is when the patient has the disease-causing organism, but they have no signs or symptoms. Next is followed by the prodromal period, a period where the patient has mild signs or symptoms, like when they stop and think, geez, do I have a sore throat? That would be the prodromal period. If you have to think about it, am I coming down with something? Mild signs and symptoms. That's followed by the period of illness, where the signs and symptoms are most severe. Usually in the period of illness is when the number of microbes is also greatest. So on the y-axis, we have the severity of the signs and symptoms, as well as the number of microbes. And then on the x-axis, we simply have the time. After the period of illness, we have the period of decline, where the signs and symptoms get better. Like if you have a fever, that would be during the period of illness. And then when the fever breaks, that would be the start of the period of decline. Signs and symptoms get better. The period of decline is followed by the period of convalescence. This is when you still have the disease-causing organism, but their numbers have been reduced below the threshold where you no longer have any signs or symptoms of disease. And then after the period of convalescence, you no longer have the disease because you no longer have the disease-causing organism. Any question about any of that?
I have a question.
Uh-huh.
During the incubation period, can you still spread the disease?
Yes. In some diseases, actually very common respiratory diseases, you are most susceptible late in the incubation period and in the prodromal period. And I'm talking about most cases of the colds and flus when you are an adult. Why you're more likely to be spreading colds and flus late in incubation and in the prodromal period compared to in the period of illness is because an adult has an immune system which kicks in by the time of the period of illness. And the immune system reduces the chance that you will spread the disease. Now, this only happens for adults, meaning children, they are most contagious during the period of illness. But for adults, they're most contagious late in the incubation period and in the prodromal period.
Okay, thank you.
Sure. Obviously, early in the incubation period, the disease-causing organism isn't high enough. It's still growing in you. And so you're less likely to spread it early in the incubation period. And early enough in the incubation period, you can't spread it at all. But late in the incubation period, you can spread disease. And that's true of COVID-19 as well. But colds and flus for adults, most contagious in the incubation period and in the prodromal period. And you can spread disease also in the period of illness. Let's see, let me say, I guess I already talked about the period of illness begins when the signs or symptoms are no longer mild. And then when the signs or symptoms start getting better, it starts the period of decline. So in what period are you not contagious? Besides very early in the incubation period, what period are you not contagious?
Period of decline.
Say again?
Period of decline.
That would be a good guess, but it's not correct. You still have the organisms in the period of decline, so you still can spread the disease. You can even spread the disease in the period of convalescence when you no longer have any signs or symptoms of disease. And this is sort of a trick question, and that is you can spread disease in every period of the disease. You can spread it in the incubation, the prodromal, the illness period, the period of decline, and the period of convalescence. And many people don't seem to understand that as long as you have the disease-causing organism, you can spread the disease. My mother used to always tell me, "Oh, I'm getting better now. I'm not contagious." And I would answer, "Mother, you're speaking to a microbiologist. I know better." You can spread the disease until you end the period of convalescence and you no longer have the disease-causing organism. And you don't know when you end the period of convalescence because you have no signs or symptoms in the period of convalescence. Now, it is true that in the period of decline and in the period of convalescence, you're less likely to spread the disease than you would in the period of illness, the prodromal period, and then the late incubation period. But you still can spread disease. And so that's something you should know. You can spread disease during every period of the disease. I should also state that when you have the cold, I do this all the time. Of course, I'm an instructor, so I can do this. Students might not have the time to do this. When I start noticing that I'm coming down with signs of a cold, I will try and get lots of sleep. Because when you sleep, you boost your body's immune system. And when I boost my immune system, I can decrease the number of microbes and the signs and symptoms, making the cold enter the period of convalescence, meaning I lower the number of microbes down below the threshold, so I no longer have any signs or symptoms, and I'm now in the period of convalescence. The trouble is, after I no longer have any signs or symptoms, I often frequently stop getting rest, And then I start getting signs or symptoms again. And I say, oh, geez, I've still got my cold. I need to get more rest. And if you get enough rest, you can make the cold move from a period of illness. I'm not sure which one, either the prodromal or the period of illness, into the period of convalescence. But you need the time to be able to get excess rest. And it only works for colds. I've tried it for flu, and it does not work for the flu, meaning it has to be a mild disease to begin with. Any questions about any of that? Now, not every disease has every period of the stage of disease. I just explained that sometimes when I get a cold, I have the incubation period and then the prodromal period. And then if I get lots of sleep, I skip the period of illness and the period of decline and enter the period of convalescence. Okay. And then there are some diseases that have no prodromal period. Like sometimes when I get the flu, at one moment I'm in the incubation period, and then the next moment I have this splitting headache. It's certainly not mild. And I said, oh, geez, I'm really sick of something. Meaning sometimes when you get the flu, it skips the prodromal period, moves from incubation to the period of illness. I'm sure that's happened to some of you. You don't have the prodromal period when you have the flu. And then there's some that skip the period of decline, the period of convalescence. And that would be something like AIDS. The patient gets the incubation period, enters the prodromal period, then enters the period of illness, and then the disease kills them. So they never enter the period of decline and period of convalescence. That used to be the case for actually all AIDS patients, but now they've come up with better drugs and people can actually end the period of illness. I'm not sure that they never throw off the disease. So they always have the HIV virus, but they don't automatically die, meaning they have a long lifespan now with modern anti-HIV therapy. Okay, but in the old days, AIDS used to be a killer, and that was just a good example that the disease tended to kill the patient, so there was no period of decline and period of convalescence. We can mention with COVID-19 that it also kills patients, in which case there's no period of decline and period of convalescence. Any question about any of that? If not, let's move on to the spread of infection. There has to be a reservoir of the infection, a continual source of the disease-causing organism. Microbes have to come from someplace, and we call it a reservoir. The reservoirs can be living or non-living. Humans are major reservoirs of human diseases. For some human diseases, the only reservoir of the disease are human reservoirs, meaning other humans. For example, AIDS and gonorrhea only come from other humans. Carriers may have inapparent infections or latent disease, so they may not know that they actually have the disease-causing organism. And with COVID-19, for example, among young people, either young adults, teenagers, children, about 50% of them can have inapparent infection. They have no signs or symptoms. They can spread disease even though they have no signs or symptoms. Any question about any of that? Animals are another living reservoir of human diseases. For some diseases like rabies, animals are the only way that humans can get the disease. If you don't know, rabies doesn't spread from human to human, so it only spreads from an animal to a human. Lyme disease, another example of a disease that we get from animals. I think Lyme disease can spread from a human to a tick to a human, but it involves coming from a tick, so it does need to come from an animal. Some zoonoses may be transmitted to human. A zoonosis is an animal disease that can be transmitted to humans. For example, rabies is an animal disease that can be transmitted to humans. Any question about that? And then there are non-living reservoirs. For example, botulism and tetanus come from non-living reservoirs. Other examples of non-living reservoirs include the soil, water, lettuce. Like lettuce and raw spinach are major sources of gastrointestinal, let's see, how do you say that? Gastrointestinalitis. I'm not quite saying that quite correct, but close enough. Gastroenteritis, there we go. Because the spinach or lettuce has a disease-causing agent, and then it's not cooked if you're eating raw spinach, and then it causes human disease. There are three principal routes by which the causative agent of disease can be transmitted from the reservoir of infection to a susceptible host. So the disease-causing organism can spread to another human, a healthy person, by direct contact transmission, such as a handshake, kissing, by vehicle transmission, like comes from the infected person to a glass, and then the healthy person uses the same glass. The infected person may spread it by the air, and then the air brings it to the healthy person. Vehicle transmission. And then vector transmission is where an animal spread the disease from an infected person or the infected animal to the healthy person. So here we have an infected person, and then mosquito bites them, carrying the disease-causing organism to the healthy person. I can't think, here we go. Malaria would be a case where somebody who's infected with malaria could infect a mosquito, the mosquito bites the human, and then the mosquito bites the healthy person. And then rabies, where an infected animal bites a human. A vector is where an animal spreads the disease to a human. A vehicle is where an object spreads the disease to the human. So with contact transmission, there's three types of contact transmission. There's direct contact transmission, and that's person-to-person transmission. This requires close association between the infected individual and a susceptible host. Some examples of direct contact transmission are like during a handshake, spreading colds and flus, kissing. I guess that would be spreading colds and flus, but also maybe, what do you call that? Cold sore from one patient to another. Indirect contact transmission is where somebody who's infected they spread the disease to the susceptible host via a non-living object. We call non-living object fomites. So a fomite is just an object. Like, for example, somebody who's infected and then they sneeze into their hands and then they touch money, and then the money can spread the disease to the healthy individual. So the money is a fomite. Contaminated tissue, somebody blows their nose and then, I don't know, discards it and somebody goes and picks it up. We can pick it up. Children, if they're infected and they touch a baseball and then the baseball is passed to somebody who's not infected and they touch the baseball and then they pick up the disease from the baseball. That would be indirect transmission from the baseball. Droplet transmission is via airborne droplets that travel less than 1 meter. Now you should realize that droplet transmission is different from airborne transmission. And droplet transmission is 1 meter to about here in this picture. And then beyond that, where it's smaller, this would be airborne transmission. And I'm not going to ever quiz you on what droplet transmission is and airborne transmission, because in my opinion, this 1 meter mark is pretty arbitrary. A sneeze, you can pick up the disease from the air within 1 meter, and you can pick it up much further than 1 meter. So after 1 meter over here, you can pick up the disease-causing organism from the sneeze. And my point is 1 meter is just sort of an arbitrary distinction. And so I don't quiz students on that. And I find it weird that droplet transmission is, let me shrink that, is contact transmission, but airborne transmission is vehicle transmission. So like I said, I'm not going to quiz you on that. Contact, direct, hand-to-hand, droplet, which we won't talk about, and then indirect transmission, like the baseball is carrying the disease-causing organism, and so the catcher will pick it up with this hand and then return it to the pitcher, and then he can, the catcher can get the disease-causing organism. Any question about any of that? All right, vehicle transmission is transmission by an inanimate reservoir, where something like food, water, body fluids, or a vehicle like an object transmits the disease-causing organism. There are actually 3 types of vehicle transmission. So there's 3 types of transmission, 3 types of direct transmission, and 3 types of vehicle transmission. The 3 types of vehicle transmission, or there's waterborne transmission, such as when contaminated sewage gets into the water, spreading the disease by waterborne transmission. And this doesn't usually happen in first world countries, but it frequently happens in third world countries. There's foodborne transmission, where somebody touches the food who's sick, and then the food carries the disease to a susceptible patient. And foodborne transmission can be when the food is undercooked or poorly stored or just unsanitarily handled. There's also airborne transmission, and that is when the airborne droplets travel more than 1 meter from the reservoir or the person to a new host. Any question about any of that? Let's see, foodborne transmission. Most of foodborne transmission is where the food is carrying the disease and there's organisms in the food. But if a sick patient were to touch the food and then someone were to eat it, that would be foodborne transmission. Vector transmission is when an animal, that's a vector, carries the disease causing organism to the susceptible patient. The most important group of vectors are arthropods, especially biting fleas, ticks, and mosquitoes. But almost any arthropod can spread a disease-causing organism. There's two ways that vectors can transmit disease. There's mechanical transmission and biological transmission. Mechanical transmission is the passive transport of the disease-carrying organism by the vector. Like the fly picks up the disease-carrying organism when it's landed on the stuff it's on. This looks like fecal matter. And the feet pick up the disease-causing organism, and then the fly lands on something like food, and then the food is consumed by the susceptible patient. Mechanical transmission, because it's passive, and the fly transmits it. Biological transmission is where the pathogen actually reproduces inside the vector. A good example of that is malaria and mosquitoes. When a mosquito bites a human, they can pick up the malaria. The malaria actually does grow in the mosquito. And there'll be a different form of the malaria, which we'll maybe talk about later. Actually, I don't think we'll talk about that in this class, but the malaria grows in the mosquito, and then the mosquito transmits the disease to another human. So that's biological transmission. Any question about any of that? Any question about the three forms of transmission? All right, if not, let's move on to nosocomial infections. Nosocomial infections are one of the most important topics that you can learn about in microbiology. And that's because many of you, if not all of you, are planning to work in the healthcare industry. A nosocomial infection is an infection that spreads and occurs in a hospital or a hospital-like setting. Another word for nosocomial infection is a hospital-acquired infection, an infection that the patient gets from their stay in the hospital or the hospital-like setting, hospital-like, like a clinic or something like that, a nursing home. And the patient did not have the disease-causing organism until they came to the hospital-like setting. So they picked up the disease in the hospital setting. It's estimated that 5 to 15% of all hospital patients acquire a nosocomial infection in the United States. That's 2 million people per year in the United States. That's also 20,000 deaths in the United States per year that occur from a nosocomial infection. It's actually the 8th leading cause of death in the United States. Any question about any of that? Nosocomial infections are seen in greater than 1 out of 20 hospital patients. It used to be around 10%, but recently hospitals have gotten a little better and now it's closer to around 5%. 50% of nosocomial infections are preventable. They have a major cost in the United States. We're not going to talk about the numbers, but this is going through it. Nosocomial infections result from the interaction of three factors occurring in the hospital or the hospital-like setting. First, there's microorganisms in the hospital environment. Hospitals are a major reservoir of sick patients, and these sick patients carry microbes. Obviously, some of the sick patients have human pathogens. That's what made them sick, and that's why the patient is in the hospital. But also, there's normal microbiota on these patients, as well as other people in the hospital. And some normal microbiota are opportunistic pathogens, and they can cause disease for a hospital patient. And then what's really bad is some nosocomial pathogens are antibiotic resistant. And that means they'll be harder to treat. Some patients get into the hospital and then they come down with a nosocomial infection. So their health decreases even further from the nosocomial infection. The clinician describes the antibiotic, and the disease is resistant to this antibiotic. The patient says, oops, that didn't work. Let's try another antibiotic. But it may be too late. The patient may die by this time. Or the second antibiotic, the disease could be resistant to it as well. All right. Any question about that? There are microorganisms in the hospital environment, and there are human pathogens in the hospital environment, as well as other microbes that cause disease. So hospitals are not a good place to be if you do not want to come down with a nosocomial infection. A second reason why patients come down with a nosocomial infection is patients in the hospital are oftentimes compromised hosts, one whose resistance to infection is impaired, either to disease, to the therapy the patient is undergoing, or to burns. Broken skin, broken mucous membranes compromise the host, making them more susceptible to disease. Burns, injections, ventilators, urinary catheters, the administration of anesthesia, all make the patient more susceptible to coming down with a disease, a nosocomial infection. Patients who have a suppressed immune system are also compromised, and the patient can be suppressed, their immune system can be suppressed by the drugs the patient is receiving, by the radiation they're receiving for therapy, and then by burns. Any question about any of that? So the hospital patient is frequently a compromised host, one who's more likely to come down with a nosocomial infection. And then there's a chain of transmission in the hospital. There are three principal routes of transmission. There can be direct contact transmission of the disease, like one patient spreads the disease-causing organism to another patient. For example, in special care units, the patients who are there have a disease-causing organism, and they're more likely to spread it from patient to patient. It can also be spread from hospital staff to patient. For example, a hospital worker can pick it up from a sick patient and then carry it to another hospital patient. There's indirect contact transmission that occurs via fomite. This happens in the hospital with urinary and IV catheters, contaminated surgical dressing. So when you're working on the dressing, you should always be careful how you're handling it so you don't contaminate the surgical dressing with a disease, particularly a nosocomial disease. And then there's vehicle airborne transmission, where the ventilation system can spread the disease. There was initially fear that COVID-19, which is airborne, could spread by the ventilation system in the hospital. And to tell you the truth, I'm not sure what the hospitals did to reduce this risk, because it isn't a real great problem. As long as you stay, 6 feet away from another patient who has COVID-19, you should be fairly safe. Another problem with airborne transmission, vehicle transmission, is when you travel on a plane. A plane is a closed environment where they just recirculate the air in the jet or airplane. And that makes a disease much more likely to spread when you travel via a plane. Any question about any of that? And that's been a problem that the airline industry is known for at least 50 years. And they've never come up with a way to solve this. Every proposal, like putting filters in the ventilation system, put in a UV light to pass the air through an area where it's exposed to UV light. The airline industry has always said this is too expensive, or it puts too much weight on the plane. Another thing they could do is just not recirculate the air, and they could take in fresh air from outside. You have to increase the pressure of it and then release it into the passenger compartment. But that would be a way that would solve this problem. And I'm not sure, maybe that proposal takes fancy engineering of the airplane, and they haven't done that one either. All right, so because you're going to be working in the healthcare facility, you should take steps to control nosocomial infections. There are certain procedures that are generally implemented to control the spread of nosocomial infections. In microbiology, you are learning aseptic technique. When you go and work in the healthcare industry, always apply aseptic technique. Always apply what you've learned in microbiology, and that will help reduce the spread of nosocomial infections. You should properly handle all contaminated material. And then when you're done handling the contaminated material, wash your hands. Frequent and thorough hand washing has been shown to reduce the spread of nosocomial infections by 50%. You should educate the staff, the patients, and the visitors. And this is something that hospitals have finally started to do. They have sanitizer outside of every room, usually in the elevator. They have a sign requesting visitors to wash or sanitize their hand to reduce the spread of disease. The education of the patient and the visitors has really helped reduce the spread of nosocomial infection in the United States. This has been in the last 10 years. There are many others procedures you can follow to reduce the spread of nosocomial infection. They have a minor effect, so I'm not going to talk about them, but you can read about them in the online chapter notes. Okay. Any question about any of that? All right. The last two slides, I want to talk a little bit about epidemiology. Epidemiology is the study of where and when disease occur and how diseases are transmitted in populations. The CDC, Center for Disease Control, collects and analyzes the US epidemiological information. Usually what happens is diseases are turned in by doctors and healthcare facilities to the local county health department. The county health department turns it into the state health department, and then the state health department turns it in the United States to the CDC. The CDC then collects and analyzes the epidemiological information and publishes morbidity and mortality weekly report each week. And because morbidity and mortality weekly report is so difficult to say that most people just call it the MMWR. And you can find that MMWR at the site www.cdc.gov. All notifiable diseases have to be reported. So notifiable diseases are those that physicians are required by law to report to the US Public Health Service. And then those notifiable diseases will eventually be turned into the Center for Disease Control. I think it takes about a week for the healthcare facility to turn it, actually it might only be 3 days, to take it into the county office, and the county office turns it into the state office, and the state office turns it into the CDC. Any question about any of that? All right. Morbidity is the incidence of a specific notifiable disease. The morbidity rate is the number of people affected in relation to the total population in a given time period. The mortality is the deaths from a notifiable disease. The mortality rate is the number of deaths from a disease in relation to the population in a given time. For example, the mortality rate for COVID-19 in the United States is now over 800,000, over 800,000. I'm not sure how much over 800,000 because I haven't looked recently, but it's over 800,000 deaths from COVID-19 over the total population. And I'm sorry, I don't know the population of the United States. Does anyone know the population of the United States? I think it's something like 6 million, 7 million, something like that. All right. Any questions about any of this? If that not, that's it for chapter 14. Let's move on to chapter 13.
Can we get a PowerPoint for this chapter, please?
Sure. Can you remind me after?
Sure. And then was the recording from Tuesday uploaded on the YouTube?
I meant to check that before the class and I ran out of time, so I'm not sure. I don't know.
I don't think it's.
Remind me of that after the class. I'll check that as well.
Thank
Because
you.
I'm not going to have time to check that now. I will try to start the login process, so I'll be quicker doing that. Ah, that's taking too long. Let me go to start the next lecture. Chapter 13. Chapter 13 talks about viruses. We briefly talked about viruses, so now we're going to talk about viruses in depth. Well, it's taking a long time to load. Don't know why it's taking so long. All right, chapter 13, viruses, viroids, and prions. This slide lists the major goals and a rough outline. Always know the terms. Like you should know what a capsid is, an envelope, what a spike is, a bacteriophage. You should know what a lytic and lysogenic life cycle is, as well as a latent life cycle, a persistent infection. That's sort of a life cycle. You should know what a prion is, a viroid, a prophage, and a provirus. Any question about any of those terms? All right. You should understand the characteristics of viruses, the structure of viruses, and the classification of viruses. Understand how viruses multiply. And then lastly, we'll talk about prions and viroids, and so you should understand about them. Any question about what we're doing? All right. Viruses replicate in a host cell. And the major purpose of a virus is to reproduce. Any questions about any of that? And how they do that is they infect a cell and then take over the cell, causing the cell to replicate the viruses. So a virus invades a cell, hijacks the host cell, and then forces it to make more viral copies, which then escape from the cell. And why isn't that playing? Oh, okay. Let me go here, the link. Are you hearing that good enough?
Yep.
Great.
In the microscopic world of bacteria, a miniature act of piracy is about to take place. T4 bacteriophages. With their spider-like tail fibers and prism-shaped heads, these viral marauders plan to hijack an unsuspecting bacterium and use it as their own metabolic nursery. This E. coli bacterium has seen its last day. Binding to its surface, the virus injects its precious cargo of genetic material into the cell, commanding it to produce a new batch of T4 bacteriophages. In a final act, the bacterium is forced to self-destruct, releasing hundreds of viruses ready to kill again.
All right, any question about that video? I think I'll close that. Are you hearing me okay? I turned down the volume with that. So
Yes.
back up. I don't know if this affects your hearing or not, but it certainly affects my hearing. Some general characteristic of viruses. When viruses are not within a host cell, they're generally inert. They display little or no metabolic activity when they are not within their host cell. Viruses require a living host to multiply. So we can say that viruses are obligate intracellular parasites. They require a host cell to replicate. They multiply within living cells by using the host cell's enzymes, the nucleic acids of the host cell, the amino acid of the host cells, ATP of the host cell. Viruses have no ATP generating mechanism. So when they need energy from ATP, they have to rely on the host cell to make the ATP. Viruses contain as their genome either DNA or RNA, but they never contain both. So the innermost molecule of a virus is its genome, which is shown here, either DNA or RNA. Now, DNA for a genome for us is normal, but for RNA, it doesn't happen in cells. But for a virus, their genome can be an RNA molecule. Any question about any of that? Outside of the innermost molecule of the virus, meaning outside of the genome of the virus, there is a protein coat surrounding the virus shown here. And this has this section removed so that we can look inside the virus. This would be a protein coat right here too. All viruses have a genome molecule surrounded by a protein coat surrounding the nucleic acid. Some viruses have a lipid envelope surrounding the protein coat, but not all viruses have a lipid envelope surrounding the protein coat. Any question about any of that? All right. Another characteristic of viruses is they all have a host range. The range of host cells the virus can infect. Usually, most viruses can infect only the cells of one or a few species. Like, for example, HIV can only infect human cells, and actually only a few human cells of humans, and then a few cells of one of the chimpanzees. Another good example would be, oh gosh, I'm dropping my memory here. Leprosy can only infect human cells and cells of the armadillo. Okay. So usually the host range of a virus is narrow, that it can only infect certain cells of one or a few species. That's not all viruses. As you know, the flu, for example, can hit humans, pigs, horses, a few other mammals, and then birds. Okay, so some viruses have a much broader host range, but most of them have a very narrow host range where they're infecting only a few cells of one or a few species. The host range is determined by specific host attachment sites, which we'll call receptors, like receptors on cell walls and flagella. And these are cellular factors necessary for viral multiplication. Usually a receptor is where the virus binds to the host cell. And then the virus will have attachment molecules for binding to the receptor. So most viruses, I state right here, infect the cells of one or a few species of cells. Bacteria viruses infect only bacteria cells. And this we call bacteriophages. Sometimes they shorten that to phages or phages. Both pronunciations are correct. I'll try and use always bacteriophage, and that just means a bacterial virus. Bacterioviruses do not infect human cells, or for that matter, eukaryotic cells. They only infect bacteria cells. When we're talking about viruses, it's important to realize their relative size. Viruses are, on average, about 10 times smaller than a prokaryotic cell. So right here, we're looking at a little bit of a red blood cell. This is an E. coli, a bacteria that's a little bit larger than your average bacteria. It's got a little bit longer. The width is about the average size of most bacteria. Excuse me. Hopefully you didn't hear that much of that cough. So viruses are smaller than E. coli. And generally they're about 10 times smaller than most prokaryotes. The virus size does vary. The smallest are around 20 nanometers in size. That's the diameter. And then they can get up to 0.44 micrometers in diameter. That would be megavirus, the virus that has the largest diameter. The longest virus can go up to 1 micrometer, and that would be close to Ebola. But that's its length. Its width is much less. So its diameter is actually smaller than megavirus. The point is that viruses can range in size up to the size of the smallest prokaryote, meaning 0.44 micrometers in diameter or 1 micrometer in length. And that's about the size of the smallest prokaryote, smallest bacteria. And then their size can get way down to 20 millimeters. The smallest virus that you're probably aware of, and most humans are aware of, is poliovirus, around 30 nanometers. But there are smaller viruses like, I'm forgetting the name of it now, bacteriophage, T2, 24 nanometers. I'm sure nobody's ever heard of bacteriophage T4, but all of you have probably heard of poliovirus. So that's the smallest virus you've probably heard of. When we're talking about viruses, it's important to realize that viruses have a structure. We call a complete virus a virion, a single mature, complete infectious virus particle. So when we're talking about a virion, all we mean is a virus capable of infecting a host cell. And usually when we're talking about newly replicated viruses, we call them virions. Viruses have a particular structure. I mentioned their innermost molecule is a molecule of nucleic acid. The nucleic acid is either DNA or RNA, but it is never both. You have either DNA inside a virus or RNA. Never do you have both. The DNA of the virus can be double-stranded, and that's normal for us. That's what we normally see in our cells. But the DNA can be single-stranded for the virus. And we do see this in our cells. When the DNA is replicating, it's single-stranded. When the DNA is being transcribed, it's also single-stranded. So double-stranded DNA and single-stranded DNA can be the viral genome. The viral genome can also be double-stranded RNA, and this is not seen in our cells. Our cells do not have double-stranded RNA, but viral genomes can be double-stranded RNA. Viral genomes can also be single-stranded RNA. When we're talking about single-stranded RNA molecules, it's important to distinguish between positive strand, single-strand RNA, and negative strand, single-strand RNA. The positive single-strand RNA molecule can be read by a ribosome. So the ribosome can read this genome, and it will read it as AUG, and then begin protein translation at AUG. The negative single-strand RNA genome cannot be read by the ribosomes. And this strand of RNA will be complementary to the positive strand RNA. So instead of having AUG, you'd have UAC for the negative single-strand RNA genome. Any question about any of that? Why is this important? Because the positive single-strand RNA genome, when it infects a cell, that genome can be read by the ribosome to make ribosomal proteins for the virus, whereas the negative strand RNA genome cannot be read by the ribosome. And then the virus will have to replicate the negative strand RNA. And when you replicate the negative strand, you make you use this as the template, you make the positive strand RNA. And then the replicated RNA can then be used by the ribosome to make the viral RNA. Any question about that? All right. Lastly, let me state that the nucleic acid viruses can be linear or they can be circular. Remember, a linear genome is what we see in eukaryote cells. They have linear DNA molecules, and then circular DNA molecules are what we see in prokaryotic cells. Of course, the viral genome can be linear or circular, and it can be DNA or RNA. And then you should note that some viruses have their genome in several segments, like the influenza virus has their genome not in one segment, but in several segments. And this should be familiar to you because humans have their genome in several segments, and we call each segment a chromosome. Any question about any of that? If not, let me briefly say that there's two other structures to viruses. There's the nucleic acid, which we talked about. There's the capsid, that's the protein coat surrounding the nucleic acid. So that's the second part of the viral structure. And then some viruses have an envelope surrounding the protein coat. And that would be the third part of the viral structure. Only some viruses have an envelope. All right, are there any questions on what we've discussed? If not, I'll stop here and we will continue here next Tuesday. If you have questions for the lab, I will be there for 15 minutes from 6:30 to 6:45. But at 6:45, I'll be logging off. All right. If there's no questions, I'm gonna log off.